Microbiome sequencing data are biased. But what if we could correct these biases? This is a question that we have been asking ourselves. My name is Luise Rauer. I'm a doctoral researcher at the University of Augsburg, Helmholtz-Munich, and the Technical University of Munich in Germany. And together with my colleagues, we have been working on extraction bias and chimera formation, which seem to be predicted by bacterial morphology and cell number in microbiome sequencing data. Let me show you how. To give you a little bit of background, whenever we investigate the microbiome via sequencing, the data are distorted by multiple biases. These biases are introduced along the microbiome data generation pipeline. And in particular, I would like to highlight extraction bias, which originates from different cell lysis efficiencies between the taxa, and also chimera formation, which is a mixture between DNA sequences of two taxa. And to study these two biases, we built quite a complex study design, which is shown in a simplified way here. So briefly, we investigated three different mock communities: an even mock community and a staggered mock community, which have the same taxa, and also a third staggered mock community, which we call the spike-in community. Of these mock communities, we had both cell and DNA mocks, and of the cell mocks, we prepared dilution series to cover high and low biomass samples. And all these dilutions and samples underwent 8 different extraction protocols, which were a combination of 2 extraction kits, 2 lysis conditions, and 2 buffers. And then we amplified and sequenced these mock communities, and we could perform the taxonomic annotation by matching against reference genomes, which were provided with the mock communities. And this also allowed us to get our first result, which is on chimeras, which we identified as sequences that match 2 different mock taxa or more. And interestingly, the proportion of chimeras increased with higher input material. And we further investigated these chimeras and found that they are mostly formed between closely related species, as you can see in the upper left part of the plot. So for example, between Staphylococcus, Bacillus, and Listeria. And in the staggered mock community in the lower right corner, chimeras were mostly formed between the highly abundant species, as you can see on the left of the x-axis. Next, we compared the effect of the 8 extraction protocols on the sample composition. And basically, we found 8 almost distinct clusters depending on the extraction protocol. And in more detail, the sample composition was mostly affected by the extraction kit and lysis condition, but not by the buffers. We further compared this extraction bias between the individual taxa of our mocks. And in this figure, the first thing that you can see is that there were extreme differences in the extraction efficiencies between the taxa and also between the different protocols that we used in this study. But what was really interesting here is that the extraction bias was also very similar between groups of taxa. And these groups were defined by the bacterial cell morphology. So you can see this on the right. We had gram-positive cocci, and then gram-positive rods and gram-negative rods, as indicated by the color of the lines. So we calculated basically a bioinformatic correction of extraction bias, and we also applied this to different samples and were able to significantly reduce extraction bias. So, to summarize our findings, we saw that high bacterial cell numbers lead to increased chimera formation between closely related or highly abundant taxa. Next, we found that the microbiome sample composition is affected by extraction kits and lysis conditions, but not so much by buffers. And last but not least, we saw that extraction bias can be predicted by the bacterial cell morphology, or more precisely by its gram stain and cell shape. So thank you for your attention, for listening. Please reach out if you have questions or feedback, and yeah, see you in Lyon.